Hello, everybody, and welcome to this live webinar on cleaning validation. It's a strange time today. We're not able to travel, not physically at least, and therefore I thought it would be a good idea that we could travel a little bit virtually today by taking a road trip, browsing through this travel guide that I made for you on cleaning validations for reusable devices. Now, it's about reusable devices. What are reusable devices? Are medical devices that are used in the healthcare facility during operations and are then processed, which means they are cleaned, disinfected, and sterilized, and then reused again in the clinical setting in the healthcare facility. So it's very important that the processing is okay. But today I will only focus on one step of that processing. It is the cleaning. The cleaning of a medical device, a reusable medical device. And why is the cleaning of a medical device important? It's important because, first of all, you don't want any soil from the former procedure to be present on the device that is going to be used for your procedure. As a patient, I would not like it. But also, you need to remove the soil to make sure that the disinfection and the sterilization of that device is possible. Soil debris will make it very hard to actually sterilize a device. Now, how do you know if a device is clean? In the healthcare facility, when they clean your device, they will only use visual inspection. So they will use a visual inspection. They will look at your device, perhaps use a magnifier, but they look at it and they will decide whether or not it is cleaned. This means that when your device is being used and cleaned, that based on the subjective visual inspection, the device is brought back into the healthcare facility, brought back into an operation. So the validation of this cleaning step is essential to demonstrate the effectiveness of the cleaning, because you have to validate and show to the authorities that when people are using this procedure, this cleaning procedure, that by using only a visual inspection later on in healthcare facilities, that we can rely on that. So the road trip today, the journey we're going to take today, is about how can we design a cleaning validation for a reusable medical device? How can we make sure that when our medical or reusable medical device has a passport, it will get a stamp in order to allow access to the border once they want to cross the border of the US market or the European market. So today I will be your guide on this tour. Let's travel. I even, for those who see me live on the camera, I've brought my umbrella. So if you see me, follow your guide, put my name tag on as an actual guide. I will guide you towards a good design of a cleaning validation. In this road trip, using this travel guide, I actually are going to highlight some Traveler Choice Awards, some things you must see on the road, some things you must take into account, and there are also some tourist traps. I'm from Belgium, and one of the tourist traps that could be if somebody says this is Belgian chocolate, and I really like chocolate, and it looks like Belgian chocolate, I pay the price for Belgian chocolate, but it doesn't taste like Belgian chocolate. That's, for me, kind of a tourist trap. So I hope we're all on board and you're ready to take this journey. So let's start. Where do we start when you want to validate your cleaning procedure? The essential documents that you need to take on your road trip is you need a cleaning procedure. Your cleaning procedure must be part of your instruction for use. It must be, of course, very easy to read, grammatically correct, and so on, but you should use images if necessary. It should be clear instructions for the healthcare facility and the personnel that is going to clean your device, what they have to do, and how that your device can be, of course, be cleaned. So if you don't have a cleaning procedure, you should look for one or create one before you start this journey of the cleaning validation. Now, how does a cleaning validation look like? The cleaning validation is, I've built it up in four main parts. It starts with the device. You're going to make it dirty. You're going to soil it, clean, and then evaluate your cleaning steps. So we start with the device. Now, each journey starts when you're going on holiday. You have to pack your bags. Now, personally, I would recommend to travel as light as possible. This is the picture of not traveling light and taking too much on. This is a picture on how I personally go on holiday, and I think we take too much stuff along. It actually slows down our journey. And do we need all the stuff? Now, when you think about your reusable devices, okay, which devices do you put in your luggage? On which devices are you going to validate your cleaning procedure? On all your devices, some of them. When you want to travel light, there is an option to group your devices into families based on the surface, the type of material, the patient context, and it's perfectly normal according to the ISO standards or tier standards that you can do that. What is the advantage? You will group your devices into families and take one representative for each family on which you're going to perform your cleaning validation. The advantages is that you can reduce the timing of all your cleaning validation. It will reduce cost. What you need is an expert input on how are you going to group your family, but also from a cleaning point of view. So you're going to need that expert input, and you're going to select worst case devices. Now, once you have your devices on which you want to perform your cleaning validation, you need to know your device. You need to look at what are the surfaces and the areas which are difficult to clean. Where are holes? Where are gaps? Where can soil intervene? Another thing you need to know is, okay, can I disassemble my device? Yes or no? Do I have to do it? And how is it used during clinical application? Patient contact, is it with skin, with blood? How does it get soiled in real life? That is what you need to know prior to starting your cleaning validation. Now, which device do you use? When you're thinking about reusable devices, you have new devices and you have used devices. What do you use in your cleaning validation? It's a very easy answer. You have to use devices that are brought in simulated use. This is a Traveler Choice Award. This is something you must do. Don't do a cleaning validation on a new device. Why? You need to bring the device into a use stage where you soil, clean, and disinfect or sterilize your device a numerous amount of time in order to allow soil to build up. We at Nelson, we use 6 cycles. You can do more, but you have to make sure that soil is allowed to accumulate to make sure that you challenge your procedure. Okay. So we have discussed our device and we know what we need from the device point. We need it in use stage. So now we can go to the second phase, is to make the device dirty, to challenge the device with soil. Which soil are you going to take into account? You need to use or construct your soil with soil components. It can be blood, it can be mucus, it can be bone marrow, bacteria, and so on. It's very important that your soil components must be clinically relevant. If you see the picture on the right, it is actually a soil made of blood and butter because it simulates the soil for this device because it's used in liposuction where fat and blood are actually mixed together. You don't need to follow always standard test soil protocols or whatever. If you think that you need to add a certain soil component because it's relevant for your device, you just have to justify why you're going to use it. You have to do it anyway, this justification, and the soil must be clinically relevant. That is absolutely important. Then you decide how you're going to contaminate your device. You have to select a worst case contamination. You have to challenge your device in the soiling, but not too worst case. For instance, in these pictures, you see the scissors. Only a part of the scissors are submerged in the soil, so not a complete device because that's too much. That's not clinically relevant anymore. And you're going to use the handles with soiled gloves, or you can spray the device. One thing you should not forget and which you should always take into account, and this is where I would lift my umbrella and say, take attention, please follow me here. You have to take into account the dry time. The dry time is crucial. You must make sure that the soil is dry. If you do your dishes at home, you know that when the dishes were from last evening, it's more difficult to clean, it's more challenging. So this brings me to the third part of the cleaning validation. It's the cleaning itself. You need to select detergents. What are the detergents you're going to use for your cleaning? How do you choose your detergents? First of all, you look at the chemical properties. Are they alkaline? Are they enzymatic based? And what is important for you as a manufacturer, you have to make sure that your device is compatible with that detergent. Your detergent should not degrade your device or destroy your device. It should be compatible. There are differences in the European and the US markets. For instance, in Europe, they prefer an alkaline detergent, whereas in the US, there is a preference for enzymatic detergents. Another thing is that you should look at the availability of the detergent on the market on which you want to put your device on that market. So if, for instance, in Europe, we don't have access to detergents and it's only available in the US, it is not a good cleaning validation and it will not give you a stamp on your passport for the US, for the European market. So sometimes it's necessary to use two different types of detergents to enter both markets. Okay, so think about the detergent, but it should be available and compatible at least with your device. Then you're going to clean your device. There are two main ways of cleaning your device. You have manual cleaning. It's really like standing at the sink, soaking, brushing, flushing. Sometimes a mechanical step is included with ultrasonic cleaners. And here I must say that removal of gross soil is essential before you use the mechanical or the ultrasonic cleaner, because otherwise your water will be easily saturated already with the gross soil and it will be very hard to clean and your validation probably will fail. But in real life, it's also not good practice. Or you can use an automated cleaning step, which is using a washer disinfector. Here, each step of your wash cycle should be clearly defined, how long it should take, at which temperature, how much detergent you're going to use, and you generally have a pre-wash, a washing, a rinsing, a thermal disinfection, and a final rinsing and a dry time step. So it's clear that you can choose between two ways or generally accepted ways of cleaning. If your device can be cleaned with a washer disinfector, you should validate a procedure with an automated cleaning. You can add a manual cleaning because not in all healthcare facilities there is the availability of a washer disinfectant, specifically when you think about reusable devices for dental applications. There, not every dentist practice has a washer disinfector. So, or the market where you're going to put your device, the hospitals do not generally have it because the budgets are lower. So things to consider, but if your device is compatible with an automated cleaning step, then you should at least validate that and ideally combined with a manual cleaning validation. Okay. What is the cleaning procedure that you're going to validate? So you have to select the worst case parameter. So on your left side, you have your IFU, your instruction for use with the parameters described there. In your validations, you're going to challenge that method by taking the worst case, which means, for instance, in the first row, the detergent concentration can be from 5 up to 15 milliliters per liters. In a cleaning validation, you will use the lowest concentrations, 5 milliliters per liter in this case, because that is worst case from a cleaning perspective. You will also use for shorter contact time, shorter duration. You will flush with a little bit less than is described, and you will use the lowest temperature. A potential tourist trap, so please pay attention, is that soiling and cleaning can be two worst case. In each cleaning validation, that is one of the most challenging part, is you have to take into account worst case cleaning and you have to challenge your cleaning parameter. When you're going to overdo this, you will actually you will fail, but it is too extreme. So you always have to find the right balance between worst case soiling, but not too extreme, and challenge your cleaning parameters, but also not to an extreme, because it still has to be relevant, and you should not always change or challenge all parameters in your cleaning step. You should take the essential ones out of it and challenge the complete set. We see a lot of failures happens a lot when you overdo it, when you're too conservative or wanting to go into the extreme. So please pay attention and find that right balance in your cleaning validation. So we have chosen the device, we have soiled it, we have cleaned it. How do we know that it is clean enough? We have to evaluate for traces of blood, of mucus, bone marrow, bacteria, and for detergent residues, right? Evaluation is done at 3 levels: level 1, level 2, and level 3. The first level is, what are we going to do is we're going to visually inspect the device. This is how it's done in the healthcare facility. So it's an important parameter and criteria also for the laboratory testing of this validation. This visual inspection is not only done at the last step. No, it is already done during simulated use, because in the simulated use cycle, you're going to use worst case soiling, worst case cleaning, and disinfect or sterilize your device and do this 6 times. And after each cycle, you should visually inspect your device, because if it fails here, you should stop already your validation, even before it actually started. Sometimes I put a nose on the slide. We have seen cleaning validation where we put the device in simulated use and everything looks clean. But the technician came to me and said, but it smells strange. It doesn't smell good anymore. Also, that shows you that something is wrong. So please broaden your mind there. If it looks clean, but it smells strange or it feels different, I would already say stop there and see what is happening before you continue, because these things will happen in the healthcare facility for sure, because there it's perhaps after 12 cycles, because it's not so worst case, but still it will happen. So level 1, visual inspection, very important parameter. Then we're going to look for traces of soil residue. We're going to take our clean device. We're going to try to remove any residue of soil which is visually not clear because it passed level 1, and see whether or not there are traces of soil still present. So we will extract the soil from the device under certain extraction conditions and analyze that. During this step of the evaluation, we will take a positive and a negative device control into account. The positive device control is a device which is soiled, so it's very dirty, but it's not cleaned, and will be used to determine the extraction efficiency. I will come back to that in the next slide. The negative device control is one device which is unsoiled and cleaned and is used as a test control to see whether or not there are interference in our analytical testing, to make sure that everything is under control. And then we will take at least 3 test devices, for which we will use for the evaluation of our cleaning procedure, which are soiled and cleaned. So that is very crucial to take the controls into account. Without the controls, you cannot conclude anything. So how do we remove the soil traces of the device? By extraction. This can be sonication, swabbing, manual shaking, flushing. And you use your positive device control to determine the extraction efficiency, because you need to know how efficient this extraction method is in order to correct your results later. That is crucial. If you don't have that information, you actually cannot make any conclusion whether or not it is a pass or a fail later on. Because once you have that extract, we're going to analyze these solutions for the presence of hemoglobin, protein, carbohydrates, TOC or total organic carbon, for bioload reduction if your soil contained bacteria or endotoxins. What is needed, and please note down, you need to select at least 2 markers to evaluate your cleaning validation. I removed 2 of them. So bioload reduction and endotoxin are not considered as one of these 2. So it can be a 3rd or a 4th marker, because sometimes it is interesting to include them, but the 2 markers should be hemoglobin, protein, carbohydrates, or TOC. And protein is the marker you should always select. And another thing is the marker should be present in your test soil. So if your test soil doesn't contain blood, then you should also not test for traces of hemoglobin. What are the acceptance criteria? The acceptance criteria can be found in Tier 30 and in the new draft of ISO 15883. And it is described in this table. So for proteins, it has to be below or equal to 6.4 micrograms per square centimeter. If it's below that, you have a pass. If it's above that, if it's 6.5, you have a clear fail. It's a fail of your cleaning evaluation. So we have already 2 levels. That brings me to the 3rd level, detergent residue. The detergent residue is, as you can see, is also important because it can also be harmful for the patient. For this testing, we're not going to clean the device with worst case cleaning parameters from a soil residue point of view, but here we're going to take 3 other devices and clean them with a high amount of detergents, like the highest in the range of your IFU. And we're going to reduce some rinsing step and see whether or not in this extreme situation, whether or not we see detergent residues, and we will evaluate it by using a cytotoxicity test. So that is level 3, the cytotox test. What do you do in case of failure? If level 1 fails, it's one of the most critical parameters because it's used in the clinical healthcare facility. You have to review your procedure. It can be a new design. It's almost always seen already during the simulated use. If the 2 markers fail, you might look at procedures, soiling through worst case. You might include an extra rinsing step or a higher detergent concentrate. You have to see, did I make my balance between soiling and cleaning correct? Can also be a new design. And if your cytotox fails, also there, did I overdo it? Did I did it too worse? But sometimes you also have to wonder, is my device already without a cleaning cytotoxic or not? We had a situation where that was the case, that the device itself was already cytotoxic even without a cleaning step, and then it was just not a good marker to look for detergent residue. We're almost at the end of the presentation, but before I conclude, I want to point one other thing. It's a little detour of this road trip, so you might still need to follow the umbrella on this detour. We're using a cytotox testing in this validation. And can these data be used to evaluate the biocompatibility? Because ISO 10993-1 says that the biological safety of a medical device should be evaluated over the whole life cycle. It has to include a reprocessing. Now, the reprocessing affects the biocompatibility by high detergent concentrations, process temperatures. When we look only at cleaning, it's the detergent, the process temperature, and the rinsing cycles. And these cycles are several times used throughout the life cycle of the device. So when we look just at cleaning today, in a cleaning validation, during the simulated use, we'll use low detergent concentration and shortest contact time of the detergent with the device. When we look at the residue testing, we will have high detergent concentration and we'll not include the final step. We will evaluate the cytotox test will be done after the cleaning. We'll not do this disinfection or the sterilization. Whereas in the biocompatibility testing, when we look at what challenges the material properties, it is high detergent concentration, long contact time, and it should include all reprocessing cycles. So you see there is a little bit of a gap between the cytotox test and how the sample is prepared for a cleaning validation compared to how somebody from a biocompatibility background will look at it. I just want to make sure that we avoid a potential tourist trap and that we just assume that the data, the cytotoxicity data obtained during a cleaning validation can be automatically used in as a biocompatibility endpoint. Can it not be? It depends. You need good justification. But if you have one take-home message, please do not just assume that. Think about it and discuss it with the biocompatibility experts and don't assume that necessarily. So this brings me to the final end of this road trip, this book, Cleaning Validations for Reusable Medical Devices. We have known or learned about the importance of a good cleaning because a visual inspection is the only way how in a healthcare facility it is decided whether or not the device is clean. We have discussed the 4 steps of the cleaning validation. I've given several awards. You can find them through the slides, but 2 for the first part and 3 for the evaluation, and warned you for 2 potential tourist traps. And with this information, you will have a passport where you will receive your stamp in order to allow access on the European and the US markets. So before I conclude, I also want to thank that I could be your guide today. And as a final tip, I would say for the US market, it's very important, or it is recommended to perform these studies under GLP accreditation, whereas for the European market, the ISO 17025 accreditation of your test facility for each of these steps is very important. So if you don't see the umbrella, which I have here anymore, you can always email me. And if you want to travel more, part 2 and 3 of this series of road trips on cleaning validation will be released. So it's cleaning validation, this is today, but disinfection validations and steam sterilization will soon come up. And if you cannot wait, of course, on our website from Sotera, so Nelson Labs, you can find all sorts of other interesting information about lab testing, or you can go to one of our sister companies, Nordion or Sterigenics, who can teach you a lot about sterilization solutions by gamma or E-beam irradiation and ethylene oxide. I believe that we still have 5 more minutes. So if there are any questions, I would love to hear from them. So you can definitely let me know in the chat or just on the microphone, but for sure. I'm not sure. Are there questions? Thank you, Ivan. If they're not today, you can always do that later, or when you're dealing actually with cleaning validations, because sometimes if you're not into this domain, then it's what are tests to identify if the cleaning? What are tests to identify bacteria if the cleaning didn't work? So if you add bacteria to your test soil, we actually would do a bioburden reduction. We will look for a bioburden reduction, but normally in a cleaning, you will actually look at the removal of soil. And later on in the disinfection study, you will see whether bacteria or viruses can be reduced. Just also for your information, we will never, or we at Nelson will never spike or challenge viruses, which is of course a hot topic today, but we will use bacteria as a representative for as well the killing of viruses. I'm not sure if I'm making myself clear there, but cleaning mainly focus on soil remover. You can determine how much reduction you have of bacteria and as a consequence also of viruses because they're less resistant. But normally that is looked at in the disinfection validation. So that's the second step in a reprocessing cycle. Or you can also already remove just all bacteria and viruses through steam sterilization, which is then part 2 and part 3. But I only had 30 minutes today, so I could not cover that. Okay. So we'll have 3 more minutes or 2, which I've been heard. Okay, then I can also wish you all a very nice day. Stay in good health, and I hope we perhaps meet at another time. And maybe I will bring my umbrella so then you can recognize me because we're now wearing always the face mask, so it will be more challenging. But the next Compamet, I will walking around there with my umbrella if I if I attend. Mm-hmm. There's a question, can we make one validation for instruments from the same material but different manufacturer? It could be if you can justify why it can be used. So if it's the same material and the same worst case scenarios, it could be. Then we have to really look and justify it. If they're the same material used in the same settings, then it can be possible. I cannot say always, but it can be. If you're the end owner of the product, then it can be. Okay. Goodbye, everybody, and talk to you later. Okay. Shut it down.